Neuron Communication by Cassandra Almond. Neurons communicate by transmitting chemicals known as neurotransmitters across the synapse, which is the space between two neurons. Before we get into details, let's go over some vocabulary. The presynaptic membrane is the part of the cell membrane that releases neurotransmitters into the synapse. The postsynaptic membrane is the part of the cell membrane that receives the neurotransmitters. The synaptic vesicles are tiny little packets that are filled with neurotransmitter molecules. Now that we have some vocabulary, we're going to look at the steps of synaptic transmission, starting with the action potential propagating down the axon and reaching the terminal button. The action potential causes depolarization, which is what allows calcium channels to open up so calcium ions can enter in. Calcium ions play an important role in connecting to and guiding the synaptic vesicles to the presynaptic membrane. Once there, the vesicles fuse with the membrane and dump neurotransmitters into the synapse. This process is called excitation. So now what? After neurotransmitters are released into the synapse, some bind to uniquely shaped ion channels along the postsynaptic membrane. These channels are called receptors. Two major types of receptors neurotransmitters might bind to are ionotropic and metabotropic receptors. After neurotransmitters have done their job, they must be removed or inactivated from the synapse. Some neurotransmitters just diffuse away, some go through reuptake, and some experience enzymatic deactivation. When a neurotransmitter diffuses, it simply is moving away from the synapse. Reuptake is when it's sucked back up into the presynaptic neuron to be recycled for later. Enzymatic deactivation is when enzymes break down neurotransmitters. So how might neurotransmitters affect the action potential of a neuron at the synapse? There are two types of neurotransmitters, excitatory and inhibitory. Excitatory neurotransmitters are neurotransmitters that increase the likelihood of the postsynaptic neuron reaching its threshold of excitation, which would allow it to generate an action potential. Inhibitory neurotransmitters, on the other hand, decrease the likelihood of the postsynaptic neuron reaching its threshold of excitation. Essentially, excitatory neurotransmitters help depolarize the neuron, while inhibitory neurotransmitters will help hyperpolarize it. These changes in membrane potential occur specifically once an excitatory or inhibitory neurotransmitter attaches itself to a postsynaptic receptor, causing stimulation. These different changes in membrane potential are specifically referred to as either excitatory postsynaptic potentials or inhibitory postsynaptic potentials. These different potentials add up to what we call summations. This occurs in two different ways. Temporal summation involves adding up potentials that occur multiple times in the same place on a dendrite over time. Spatial summation involves adding up potentials that occur at different places on a neuron's different dendrites all at the exact same time. So how does this all work together? Temporal summations of excitatory postsynaptic potentials makes it more likely to reach the threshold for an action potential if excitatory signals arrive quickly enough. But temporal summations of inhibitory postsynaptic potentials make it harder to reach the threshold. When it comes to spatial summation, the multiple excitatory postsynaptic potentials all at once will increase the chances of an action potential, while multiple inhibitory postsynaptic potentials all at once will reduce the likelihood of an action potential occurring.